Alright, and this is going to be the last, well, not half of the chapter, probably last third of the chapter. It'll be a little bit shorter lecture, but I think it was important to divide it up. So, picking up where we left off, let's talk about denaturation of proteins a little bit, and then we'll get into some of the subtleties of folding, and the process of folding. So, we've talked about denaturation prior to this, but it's worth revisiting and going a little bit further into what denaturation really is, and how different environmental effects, whether it be heat or salt or pH, affects or can drive the unfolding and loss of function of a protein. So we can think of denaturation as the changing of the tertiary structure of a protein due to these environmental influences, or we can consider denaturation something that we measure as a loss of functionality. So it's two really different approaches. One is thinking of denaturation strictly as the changing of the tertiary structure, which is less than ideal for biological function, or we can go straight to the functional component and say denaturation can be measured as a result of loss of function of a protein from its intended process or intended function biologically. So it's important to stress that the proteins in their environment, which they're suited to function, have a really fine-tuned structure that's only marginally stable. And this stability, this fine line of stability, is achieved through a suite of weak force interactions that dial in that very, very specific tertiary structure. Now, if we alter the environment by supplying more energy to the system, which increases the ability to disrupt some of those weak bonds, and that's in the form of heat, or we can use chemical treatments such as salts or pH, which change the way the solvent interacts with the surface of the protein, or changes the way in which the protein initially collapses in its folding process. All of these subtle changes can result in a different preferred tertiary structure, and that different structure is considered denatured from the native functional protein. So our most basic example of this is cooking, because we do this as a mechanism to make food more, well, arguably more delicious all the time, and there's also other benefits, including increased nutritional availability and various other things. But we can just view it from a cooking perspective. Eggs or albumin proteins in eggs being cooked on heat. Now, if we were to do an experiment, we could get a similar discoloration and denaturation by treating the egg yolk or egg white with concentrated hydrochloric acid. You would rapidly see a similar discoloration where you get a firming of the protein where it cross-links and turns opaque instead of clear. Conversely, you could get a different type of denaturation if you were to treat the egg white with concentrated sodium hydroxide. It would become more gelatinous, but not turn white. So there's a variety of different outcomes from the different chemical treatments that can lead to denaturation. All denaturation is not a process which ends in the same outcome, except for the loss of function. So the next couple of slides are just slides which indicate that a denaturation does result in loss of function, and we can plot this degree of loss of function to temperature or change of chemical environment. So in this particular case, we're just looking at the fraction of native protein in its native state, and as we increase the temperature, right around 50 degrees Celsius, you rapidly start to see, let me grab a pen, right around 50 degrees Celsius, we see a rapid decline in its loss of structure and loss of function, so that at about 70 you no longer have any of the native protein structure function remaining. Likewise, if we view it not from a heat perspective, but through a denaturation due to guanidine hydrochloride or urea treatment, you can see as we add, let me re-grab this, increase our levels of salt to solution, right around 2 molar, we start to see a increase in the unfolding of the protein, and then we have a nice plotted trend all the way up to about 6 or 5.5 molar, where we no longer have any natively folded protein. It's all unfolded. So let's talk a little bit about the mechanism of protein folding. We've been doing a little bit with it, but it's worth, like I said, like everything, revisiting in more detail. So how does a protein fold? Well, when we are talking about conformations in organic chemistry, we talk a lot about random sampling around free rotations. And thermodynamically, the tendency is for whichever conformation is the lowest in energy or most stable to be preferred over the higher energy systems. Doesn't mean that it can't exist in the higher energy system, it's just that it prefers the lower energy conformation. Now, the difference between that and the folding in proteins is that proteins have many, many, many more points of rotation. And it's a much more complex question than some of the earlier examples we viewed from an organic chemistry class perspective. And one of the things that's worth discussing is Levinthal's paradox. Levinthal's paradox is just a demonstration that proteins cannot fold in biological timelines through random sampling alone, in thermodynamically driven random sampling. And there's a couple reasons for this. One, it just would take too long. Two, there's some biological confounds because proteins don't fold in a vacuum. They fold as they come off of the ribosome, and they're in a very macromolecule dense environment, which can lead to nonspecific interactions. But most importantly, if you model protein folding entirely based on random sampling from a linear primary structure, you'll find that you'll get stuck along wrong paths, because the random sampling from a linear structure can result in you going down the wrong direction a little bit and then getting stuck in a little bit of a hole, energetic hole, which is hard to come out of through random sampling. So there's all kinds of issues with the random sampling model. Now, what this means is that there has to be some kind of specific folding pathway or order of events which makes the folding of complex proteins more efficient in terms of speed and in terms of specificity, getting the right outcome. So, what are some of the factors that come into play here? The most commonly held hypotheses at this point is that there's an order of events to protein folding, which includes firstly forming secondary structures. So, secondary structures form rapidly through hydrogen bonds due to hydrogen bonding between relatively proximal amino acids. So they can form quickly without having to change the tertiary structure significantly or have to randomly sample very far to get to that point. And as they start, they collectively drive the process. So the first step is you rapidly form alpha helices, beta sheets, and likely the turns. And this is probably the first step, which happens very quickly. Once you've achieved all those helical and beta sheet structures, you significantly reduce the degrees of freedom associated with the structure, and random sampling is a much more simple problem than it was before, but it's still a significant problem. What happens next before random sampling becomes in play is the hydrophobic collapse. Now, the hydrophobic collapse is the term which describes the coalescence of all of the nonpolar residues towards the center of a globular protein. Meaning that once you have all of your secondary structures established, all of the nonpolar residues are going to try to hide from the solvent. And this is important to remember because the solvent will try to organize itself in clathrates to balance out interactions with those nonpolar residues, which is not favorable in terms of the system's entropy. So to reduce that effect, all those hydrophobic residues collapse to the center as much as possible. And this is going to significantly reduce the freedom of rotation further, significantly reduces the complex of the thermodynamic sampling available to the protein further. And then, following this hydrophobic collapse, there's likely a rapid formation of long-range weak interactions between these secondary structures, where we have some hydrogen bonding, where we have some ionic interactions between charged residues, etc. Things like this, which will give us further refinement and even further reduce the degrees of freedom in the structure that are subject to random sampling. So, once we've reached this refined collapsed structure, we refer to this as the molten globule. And the molten globule is really the place where you can have multiple subtle differences which change the structure of the protein between a number of different similarly energetically favorable states. Doesn't mean that there isn't a preferred state, but there's a lot of similar states at this point. And so this is where a lot of that random sampling, thermodynamic sampling can take place and refinement can take place. It turns out I actually just talked about this in pretty good detail. So just to reiterate, what drives protein folding? What drives protein folding is the chemical characteristics of the primary amino acid sequence, which dictate secondary structures. Following the formation of secondary structures, you get a hydrophobic collapse in which chains which are hydrophobic bury themselves as far away from the solvent as possible, exposing the hydrophilic residues to the surface. And also it's worth mentioning once again that there's a tendency to undergo right-hand twist due to the L-amino acids. So this is a nice diagram of this process. It was originally proposed by Kendall, and the idea is that you can think of protein folding as a funnel, where a three-dimensional funnel in this case, where we have on this axis increasing energy. So as we go down the funnel towards our most stable structures, these are the lowest energy, and where we have our highest energy systems, we have our least stable structures. Now, obviously, what's lovely about this, if you were to do this as kind of an integral, you would see that at the top, we have an enormous variety of different outcomes at that energy level. But as we form secondary structures that then collapse into a hydrophobic collapse system, which then undergo refinement due to short-range interactions into our molten globule, our degree of possible outcomes is reduced significantly. So it's a really elegant way to view this funnel or cascade of protein folding. What I also like about this is that it indicates some little speed bumps along the way. These are these energetic issues that we talked about, where you could find a minima real easily and get stuck here and have a hard time climbing back out of that minima if you were just doing this via random sampling. It's very common for me to ask a question pertaining to the driving forces associated with protein folding. And most of the points associated with this question have to do with entropy of the system. By far, the most important single factor following the rapid formation of secondary structures via proximal hydrogen bonding is the favorable contribution of reducing the organization of the solvent around the protein. So if you expose hydrophobic residues, you're significantly reducing the entropy of the system, and that is not a favorable process. So to gain thermodynamic favorability, the protein is driven to hide the hydrophobic residues as part of the hydrophobic collapse. So, to reiterate, nonpolar residues force order of the solvent while unfolded, and the folding of the protein reduces that ordering, and that is what is most important following the formation of secondary structures. Now, a couple times I've mentioned marginal stability, and I think it's real easy because we think of chemicals as stable, and proteins are stable in the sense of their primary peptide bonds linking each amino acid residue. But in terms of the tertiary structure, they're only marginally stable. So, what do we mean by marginal stability? Well, this is a nice little image down in the corner here, where you can see a lot of different vibrational modes of all of these different R groups on this folded peptide. There's a lot of flexibility in this system, and this is incredibly important because this flexibility is essential for its function. A stable protein means that it doesn't want to change. Something that's too stable will be too rigid and inflexible. And because protein structure function requires change or pliability for function, something that's too stable is going to cost too much energy to actually function. So you need a protein that's just right without a ligand bound. When a ligand associates, that causes a changed structure, refinement of that tertiary structure, which drives the change. So that motion of the protein is actually essential for the chemical processes that it's facilitating. It's also important that we can regulate and turn proteins on and off, because we don't want them on all the time. We would lose control metabolically. So it's essential not just for energetics in terms of structure function, but it's also important for control, because we need to have a death grip on our metabolic controls. Willy-nilly chemical happenings in part of biology is not a good thing. So, the flexibility of protein is really important for all kinds of functions, which include ligand binding, which I mentioned, enzyme catalysis, which we'll get into significant detail in the next chapter, and regulation. And I guess it's worth just reiterating, this is important for energetics, meaning an overly stable protein will cost too much energy to change between conformations, and it's important for control. We need efficiency and we need control, and these are things that I actually questioned people on in the last test, and it's a point that keeps made probably in every test. Biochemistry is about energetic, efficiency, and control. So, let's zoom out a little bit from folding mechanisms and look a little bit more at higher level organization of proteins. So, if we look at the structure of globular proteins, even though they look like globules, you'll see that there's actually inherent structure. And we started to touch on this a little bit in the last lecture, where I talked about this core, more rigid core, which then had some structure surrounding that core, which supported the outer surface of the protein where the chemistry is taking place. And this is a more detailed look at that. So, even though we have hydrophobic residues buried in the core, that doesn't mean that there aren't planar systems in place. So if we look at this first case with cytochrome, let me grab a different here in the middle on A, you can see that we really have two different layers or planar systems. And this is really important because we're talking about freedom of rotation, right? So we have this little undefined linker down at the bottom, and we have a little linker at the top. And we really have the ability for these to slide relative next to each other. And that's the type of rotation these are going to have. This is probably refined a little bit by a hydrophobic interaction between these helical structures, which is hiding from the outside. So as they slide, they're actually motivated to associate as well. So it's not free sliding. It's just that's the planes moving next to each other. And it can get a little bit more complex. We can have a system like this kinase in B, where we have a couple different planes around a core. And it's not uncommon to see 2 or 3 planes. It's less common to see 4 different planar structures on a single globular protein, but it does take place. So this phosphorylase, C, has 4 different planes around a core. And this is just a way you can visualize the structure of a protein and give you a little bit of insight into how the core of the protein can move during conformational changes. Now, once again, sometimes people see a figure like this and they think immediately, oh no, I have to memorize this. And you don't have to memorize this figure. This is just a demonstration of the 4 different general classes of globular proteins in terms of how they're organized. So you can have proteins which are entirely composed of alpha helices. And that's what we're looking at on top, are alpha proteins. And these are just helical structures exclusively with short linking chains. We can also have beta proteins, which are entirely composed of beta sheets. And that's what we're looking at with yellow. And then we've got alpha-beta proteins in which the helical structures are intermingled with the beta structures. And we also have alpha plus beta, which are different from alpha-beta because they have separate alpha helical domains and beta domains. So alpha-beta proteins, the domains are intermingled, where the alpha plus beta, you have separate domains. So you can really see that here. Let me grab a different, something that isn't yellow or red. You can see here you have a separate beta domain from your alpha domain. Here you have a separate beta domain from your alpha domain. Unlike here where your beta and alpha domains are clearly intermingled. So something that we need to talk about a little bit is the final refinement or non-ideal folding of proteins. Because even though the formation of proteins, tertiary structure, secondary structures and tertiary structures are driven by thermodynamics, by proximal hydrogen bondings and hydrophobic collapse and formation of the molten globule, there's a lot of ways that can still go wrong. And biological systems have to have some degree of fail-safe to try to make sure, after investing all of the energy of making a peptide chain, that it comes out right. And another reason that we have biological systems that help refine protein folding is because the chemical environment in which a protein is synthesized and folded is fraught with other large macromolecules, which can lead to a host of non-specific chemical interactions, which can interfere with the protein's folding. So we have what's called a chaperone protein. And chaperones were originally identified as heat shock proteins. That's what I was introduced to them as, is heat shock proteins. And that's because the protein was identified as being upregulated following small organisms being exposed to a heat shock. And that caused a lot of denaturing events, so the organism upregulated its heat shock protein production to help fix the problem associated with that heat shock and all of the other proteins being synthesized. So, to reiterate, this is to protect the cell against the particular environment in which proteins are being folded, which are protein-rich in the matrix, and to prevent some aggregation of proteins as they're in the early stages and the hydrophobic residues are not collapsed, and possibly is hypothesized to increase the speed or accelerate slow steps in protein folding, get them through some of the sticking points. Now, it is important though, chaperones don't come for free. They are ATP driven, which means when we utilize them, they do come at an energetic cost. So this is a simple diagram of heat shock protein 70 or HSP70 chaperone doing its thing, where the heat shock proteins approach the protein as it's coming off the ribosome and help get to a folded state which is correct. This can also involve the insertion into a chaperonin complex, not chaperone, chaperonin complex, which can lead to further refinements and get you the rest of the way in the folding process. And if we want to zoom in a lot and look at how complex and elegant the chaperonins can be, this is a crystal structure of the chaperonin proteins from E. coli, and a little indication of how ATP can come on in, allow protein to interact, get capped, get refined, more ATP, causes a release and a resetting, so it can do it all over again. I really like this figure because it illustrates how chemically complex the environment protein synthesis is. When we're talking about proteins moving down this thermodynamic funnel, everyone's probably imagining something floating in solution, devoid of other interaction, having time to randomly sample itself thermodynamically to a perfect place. And that's just not the case biologically. Protein synthesis takes place along membranous systems. These systems are very densely packed with proteins and lipids and modified by carbohydrates. And there's an enormous amount of macromolecular crowding and very, very high physiological concentrations in that particular area of these different macromolecules. So this can lead to a lot of nonspecific interactions. And this just gives you an idea of how dense this really is, where the orange illustrations here are proteins which are undergoing folding, and then we have this sort of chaperonin protein with the dark green. We have a couple different, we have purple is our ribosomes, which is really nice right here. You can see the protein coming off the ribosome. Salmon is our RNA because that's also part of the ribosome. Now, we spent a lot of time talking about proteins so far, but we've been focusing on proteins which are the most stable versions of proteins, because they're the most simple examples to start with. But not all proteins are refinements. there's a lot of room for small modifications to make large differences. And some proteins don't have well-defined structures until they interact with the substrate that they're designed to interact with. So to kind of go down that direction of discussion, many proteins do exist as ensembles of structures. You can have a protein that can dramatically change its structure with small modifications. And a neat example of how significantly a single chemical change can impact a protein is in this figure where you're looking at the conversion of a leucine residue at the 45th position to tyrosine. And this is those red residues here and here. And the integration of the tyrosine in substitution of the leucine completely changes not just the tertiary structure, but the secondary structures as well. You can see in the leucine containing protein, you have 3 helical structures. And then once you've replaced the leucine with the tyrosine, you have 3 beta sheet structures and a single helical structure. What's also really neat about this is you see some ring-ring stacking, some anti-parallel ring stacking between 2 aromatic residues because of this. So this interaction here, this ring-ring stacking, is likely driving a complete restructuring of the secondary structures and therefore the tertiary structure. So this is talking about inherently or intrinsically unstructured or disordered proteins. So IUPs have no well-defined tertiary structure. They're almost always highly soluble. They're nearly always full of polar residues and lacking in any repeating hydrophobic residues which will lead to any kind of collapsed structure. And until they make contact with their substrate that they're designed to interact with, they don't get a defined structure. So what it looks like when that happens is like this, where the yellow strands are our intrinsically unstructured proteins, and you can see how they gain structure by interacting with different target proteins. Now, it's not uncommon to have questions associated with the kind of pathologies associated with protein folding, or more specifically, protein misfolding. And they're prevalent. Alzheimer's and Parkinson's are diseases of protein misfolding, which involve plaque formation, which lead to chronic inflammation and apoptotic signaling of the neurons. Cancer is technically associated with protein folding, even though it's multivariate and very complex. The p53 protein has been indicated as being a major player in the formation of lots of different cancers, and misfolding of p53 is one of the factors associated with the development of a lot of cancers. There's cystic fibrosis, which is another example of a misfolded protein leading to a terrible biological health outcome. So let's finish with talking about quaternary structure a little bit. So quaternary structure is what happens when you get a couple different tertiary structure subunits which interact with each other, forming a more refined, larger protein system. And here we're looking at the alcohol dehydrogenase enzyme, which is prevalent in the liver. And since we're coming up on spring break, I think everyone can appreciate that we have this, hopefully. And what I'd really like for people to focus on is the fact that imagine each of these subunits separately in solution. Their hydrophobic residues would be buried in the core, and they'd be soluble subunits. When they interact with each other, those hydrophobic residues are going to move from the cores of the proteins, tertiary structures, towards the interface. Because as they interface, you're developing a new core for the protein. And this association is what's going to keep these subunits associated with one another through weak chemical interactions. But that new association, moving those hydrophobic residues from the cores towards the new core, is going to cause a significant change to the structure function of the surfaces and cause a significant refinement to the chemistry at the surfaces of these proteins. So the quaternary structure can involve spatial organization of a series of reactions where just by spatially locating a reaction you can get efficiency going from one step to another step to another step along a multi-protein system. However, it's not just about spatial efficiency, it's about making each of these subunits active because of their association. And once again, this is very favorable because of the gain in entropy due to this organization. If you have two small spheres in solution, that's much more surface area than if you put them together. So putting them together has an energetic advantage. And you can get a lot of variety to how proteins organize in quaternary structures. You can have varieties of proteins which have quaternary structure due to pairing of just 2 subunits, but we can also have 3 subunits or 4 subunits or 6 plus 6 of the same, or as many as 180 subunits. There's an enormous amount of variety in quaternary structures, and to kind of point to that, all these are multimeric proteins with complex but symmetric quaternary structures, and I think this is just something that people can appreciate how complex and structurally beautiful these actually are. And that's it for the chapter 6 here. I just wanted to give everybody another case study. This is a really, really good case study to work through, which gives you some insight into protein folding as it pertains to a couple different modifications to proteins and how it might affect things biologically. So go ahead and give this one a run through. It's a short description, has a couple questions associated with it, and I think it's worth everybody giving it a little bit of time, because this is one of the classics, and like the other one, even though the other one didn't show up because of time constraints on the test, these are fair game. So go ahead and give it a look-see and see what you can come up with. And that's it for this chapter. So hopefully you'll be able to get going on the enzymes here in a little bit, and we'll have a couple more lectures up over the next couple of weeks.